लर्नर्स, आप सभी का एक बार फिर से स्वागत है हमारी इस वीडियो में। आज की वीडियो में हम पढ़ने वाले हैं प्रोडक्शन ऑफ ह्यूमन इंसुलिन बाय यूजिंग रिकॉम्बिनेंट डीएनए टेक्नोलॉजी। कैसे आप आरडीएनए टेक्नोलॉजी की मदद से ह्यूमन इंसुलिन का प्रोडक्शन कर सकते हैं, ये हम स्टार्ट करते हैं। मैं आपको बता दूं कि हमारा जो ह्यूमन इंसुलिन है, इसको हम ह्यूमुलिन के नाम से जानते हैं और ये एफडीए के द्वारा अप्रूव्ड फर्स्ट बायोटेक्नोलॉजिकल प्रोडक्ट है। ये बनाने वाली कंपनी जो थी वो थी आपका इली लिली फार्माटेक। इस कंपनी ने आपके ह्यूमनिल नाम के प्रोडक्ट को बनाया था। इंसुलिन का आप सभी को रोल पता है। इंसुलिन हमारी बॉडी के अंदर बहुत ही इंपॉर्टेंट हार्मोन है। बात करते हैं हार्मोन नेचर की, तो ये आपका 51 अमाइनो एसिड से मिलकर तैयार आपका एक हार्मोन है। इस 51 अमाइनो एसिड्स में आपका ये जो 51 अमाइनो एसिड्स है, ये एक दूसरे के साथ दो चेन्स में आपका अरेंज रहते हैं, ए चेन और बी चेन। ए चेन जो होती है वो बनी होती है आपका 21 अमाइनो एसिड्स से, और आपकी जो बी चेन होती है वो बनी होती है आपका 30 अमाइनो एसिड से। 30 अमाइनो एसिड्स कंटेनिंग बी चेन एंड 21 अमाइनो एसिड कंटेनिंग ए चेन, those are connected via disulfide bond. So disulfide bridge, it helps to join the A chain and B chain together, and it stabilizes the whole insulin structure molecule. Moving on, it produces from our pancreas. There is a leaf-like structure just beneath our stomach. It has two kinds of cells, alpha and beta. So beta Langerhans produces insulin hormone, which has a role to reduce the sugar level in our blood. How it reduces the sugar level in our blood? Actually, whenever the glucose reaches to the higher level in the blood, so it increases the uptake of glucose by the adjacent cells. There are number of cells nearby the blood pool. So whenever there is a glucose in the blood, every cell need energy in form of glucose. So they take up the glucose from the cells. So now what happens? Insulin act in order to increase this uptake of glucose from the cells by the adjacent cells. This is how what happens, the elevated or the increased level of glucose in the blood comes to the normal level. So this is the basic function of insulin. Now there are different reasons for not functioning the insulin properly. The first reason, there might be the destruction of the beta cells. If there are no B cells in the pancreas, there will be no production of insulin. Sometimes there are some genetic faulty genes which are responsible for not producing the insulin hormone in our body. So as a resultant, glucose level will not get normal when it reaches to the elevated level. So that these are the scenarios where the insulin plays very important role. Other than the regulation of glucose in our blood, insulin also helps in the metabolism of lipids and proteins as well. At the same time, it also helps in the DNA synthesis. So now you can understand the importance of insulin. It also plays very important role in the metabolism also. In metabolism, there are two kinds of mechanisms, catabolism and anabolism. Catabolism is a breakup process where the food gets break down, and on the other hand, and in anabolism, it's a build up process. In this, the products build up, it forms something. So insulin, on the one hand, it stimulates anabolism, okay, and on the other hand, it inhibits the catabolism pathways in our body. So these are the basic roles of insulin in our body. Now moving on to the production of insulin. Now, as you all know, if our body is not able to produce insulin, what is going to happen? Our glucose level will get increased. Eventually we will be suffering from diabetes, the sugar condition, or the hyperglycemia condition. So in order to treat the diabetes patients worldwide, we need a commercial production of insulin. So in order to supply this commercial amount of insulin to the worldwide patient of diabetes, the pharmaceutical companies always try to manufacture insulin in bulk quantity, which the insulin should be very safe, effective and stable. These are the three challenges for any pharmaceutical manufacturer to produce insulin. The insulin should be stable, it should be effective, and at the same time it should be stable also. So the first comes conventional insulin. As you all know, conventional insulin means the insulin which was prepared from the animal pancreas crude extract, in which zinc was added, which helps to stabilize the insulin structure, and then it purifies, then recrystallizes, and then it marketed or supplied for the human consumption. But as you all know, it is produced from the animal pancreas extract, so it might be immunogenic in nature. There were two animals, there were two kind of insulin produced as a conventional insulin. The first was bovine and the second was porcine insulins. Okay, so in porcine insulin, the pig was taken, and from pig the pancreas was removed, and then from pancreas the insulin was extracted out, which is then again further purified, recrystallized, and then supplied as purified insulin product to treat the diabetes in humans. But later on it was found to be little immunogenic in nature. Okay, but so this was the conventional insulin. But there was one issue with the conventional insulin, that this insulin was dependent upon the supply of meat, but it is not necessarily proportional always that the diabetes patients worldwide will be proportional to the meat that we got from the animals. So there is not a particular assurance of supply of this animal product. So now the scientists or the researchers from the company like Genentech and Eli Lilly, they have come forward to produce the concept of rDNA technology. As you all know, recombinant DNA technology. So what they do actually, let's start what they do in this process. So first of all, I will tell you about, let me rub this. There was a company called Genentech Pharmaceutical Company. They have taken a nucleotide sequence which codes for A and B chain of insulin, and then they introduced this nucleotide sequence into a gram-negative E. coli bacteria. So now when this nucleotide sequence entered into the E. coli bacteria, E. coli translated this nucleotide sequence, and then it started producing a lot of hormone, lot of A chain and B chain of the insulin hormone. So now we culture. What they do after that, they start adding the culture medium into the fermenter, so that the large amount of E. coli can be grow there, and large amount of insulin can be produced by n number of E. colis. After culturing in fermenter, they have got A chain and B chain in the fermenter. Now we have got the structure of insulin, A chain and B chain. Now what we need to join these A chain and B chain via the disulfide bridge. So what we do now, we perform this fermenter under suitable oxidizing condition. Under suitable oxidizing condition, this fermenter starts making the disulfide bridge between the A chain and B chain. So what happens? The disulfide bridge forms between A and B chain, and the structure of insulin completes itself and insulin is produced. Now this obtained insulin is again performed several tests, like there was a gel filtration in order to remove all the other products along with the insulin. Like there might be some contamination due to the E. coli derived products. So we need the stringent purification of this insulin obtained from our DNA technology product, because there might be some derivative products from E. coli which might be immunogenic to human being. So we need the stringent purification and chromatographic methods in order to get the pure insulin product. So we do gel filtration, we do reverse phase HPLC, and then we perform different procedures like X-ray chromatography, different procedures to make sure and to know that this produced insulin is identical to the human insulin. And this is how they produce insulin, which they call insulin CRB. They call this insulin CRB. Now this insulin CRB was very popular among all of the people because of its reassurance of supply. Okay, so there are basic three advantages of our DNA technology produced insulin. The first was reassurance of supply. There will be reassurance of supply, first advantage. Second advantage, there are less or no chances of accidental transmission of disease from any animal pathogen, because here we are not using any animals. The third advantage was this was economical process. In the long term, once you need capital to invest in this process to develop, but later on in the later stages, it produces very economical results. Now, in the current scenario, more than 10,000 gallons of fermenters are continuously being employed for the production of human insulin by using rDNA technology. So this was the human insulin CRB. Later on, there was another pharmaceutical company called Eli Lilly. So what they do, it is around 1984. It is somewhere 1984. So Eli Lilly, what they do actually, they took a nucleotide sequence which codes for proinsulin. They did. So as we have studied till now that what was insulin CRB. Insulin CRB was manufactured by the Genentech pharmaceutical company. Moving on to the another well-known company, Eli Lilly. So Eli Lilly have got idea, why not we introduce the nucleotide sequence which codes for the proinsulin. As I told you, proinsulin is the inactive form of insulin. Upon action of proteases enzymes on proinsulin converts itself into insulin. So what they do actually, what was their method? They took a nucleotide sequence code for proinsulin and they introduced this code into the recombinant E. coli gram-negative bacteria. And they provide certain cultural medium so that this E. coli can replicate itself into n copies of it and can produce n number of large amount of proinsulin. So what happens after the culturing process done in the fermenter, n number of E. coli were there, they produce proinsulin, and then under the certain suitable oxidizing condition, the proinsulin was obtained, and this proinsulin was then purified by using several methods like gel filtration method and then ion exchange method. After filtration, we got the purified proinsulin. After confirming the identicality of this proinsulin to the exact natural proinsulin, by performing reverse phase HPLC, we got the chromatogram which was identical to the human insulin. So we got insulin, and then we add into proinsulin a proteases enzymes which break the C-peptide bond in the proinsulin and convert the proinsulin into insulin. And then it was again subjected to the reverse phase HPLC, which shows the chromatogram of your newly synthesized insulin with the standard insulin, and it shows that these two chromatograms are identical to each other, which means that जो insulin हमने produce किया है by using this method, it is the identical to human insulin. That means it will perform the same function as the natural human insulin is supposed to perform. So now we can say that this process is very interesting and very applicable because of its reassurance of supply. We are not harming any animals. We are not dependent on the meat supply of animals to produce or to satisfy the human needs. So I hope आपको ये वीडियो समझ में आया होगा. अगर आपको कोई डाउट्स है तो आप कमेंट सेक्शंस में मुझसे डाउट्स पूछ सकते हैं. थैंक्स फॉर वॉचिंग माय वीडियो.